And I'm so delighted to be here, and I have to say, I don't think I've ever presented in a room with such a beautiful view. It's sensational. I'm here today to tell you all about our next generation therapeutic biologics company, Elasmogen, and the domains that we're developing that we call Sullamurs. So Elasmogen really is focusing on anti-inflammatory disease, particularly eye disease, which I'll tell you a bit more about in a few slides' time. But we see this as a gateway opportunity. It's already a £3 billion market for uveitis, which is our primary indication, but it opens out into a much larger £90 billion market for anti-inflammatory disease, such as IBD, and we're already working in IBD, and we have some data to support that. The company has succeeded in securing collaborations also in oncology, and we're working with Almac Discovery right now, creating Sullamur drug conjugates, which is a really exciting area. We have a comprehensive and robust IP portfolio, so we've protected the platform, the process, but also the products. We have a great scientific and management team in place, ex-biotech, ex-pharma. They know drug discovery, they know the field. And although we're a relatively new company, we just spun out this year in February, we've already managed to secure £2 million in non-dilutive and £850,000 in seed funding. We're out here at the moment doing the circuit, and delighted to be here as part of that process, looking for £10 million in Series A funding. So what are Sullamurs? Well, they begin their lives in sharks, believe it or not. They are the smallest naturally occurring binding domain out there. They're part of the adaptive immune system of these animals. And although they're only 11 kilodaltons, they pack a punch. They have 4 loops, which creates great diversity. And they have an interesting topology or shape, which enables them to bind into cryptic or hidden epitopes. They are incredibly stable. We can take them down to pH 1.5, they'll still bind target, or we can boil them and we can leave them for an hour and they will still bind target. They're quite incredible. They are non-glycosylated, so from a manufacturing perspective, we can go eukaryotic or prokaryotic, no problem at all. And what's also, I think, really critical to communicate to you is that although they look like an antibody, they act like an antibody, from an evolutionary perspective, they are distinct. So we get the benefits of avoiding that complex IP minefield that's out there surrounding the antibody world. So the anti-inflammatory market, as you're very well aware of, is very large, but importantly, it's growing. Right now, steroids are really the mainstay therapy, but interestingly, about 30% of patients don't even respond to steroid therapy, and it can do damage in the long term. Hence, there's been a shift in market interest to biologics, the ability to target and hone in on disease targets specifically and avoid off-site toxicity. And these guys that are out there at the moment, like Humira, for example, are targeting large opportunities like rheumatoid arthritis. But they're not tailored for specific anti-inflammatory diseases. So we're looking at uveitis, for example, and there's simply not a biologic out there. So Elasmogen is stepping aside from the competition. What we're doing is we're designing site-specific biologics to really address these unmet medical needs, these orphan diseases. We can isolate our Sullamur domains from 2 different platforms, immunization and very large synthetic libraries. We can isolate them against anti-inflammatory targets, and we've already shown that we get efficacy in rheumatoid arthritis. And that's what the images there on the left are. So you can see the deep staining, which shows inflammation, chronic inflammation, and you can see with our domain there on the left-hand side, a clean joint. So they work. And we could go for rheumatoid arthritis, but it's a very large marketplace. And if you're starting with a biologic that's so small, selective, specific, with high affinity, why not change the approach? Why go for a different route of administration? So we're going for site-specific delivery, and we're going for eye disease because it fits beautifully with the ability of our domains to cross over the cornea. Our primary indication is uveitis. It's a devastating disease with a rapid onset. About 120,000 patients worldwide will suffer from this disease, and they are refractory to current therapy. Steroids, as always, step up, but 30% of patients don't respond, and continual use of steroids causes damage. Our projected market is £3 billion, and that's based on discussions with clinicians who see reimbursement costs at about £25,000 per patient. And the reason for that really is that this doesn't affect just the elderly, it affects working stage people. So from kids all the way through to old age pensioners. And because it's a 120,000 patient group, it's an orphan indication, which is great from a regulatory perspective. So we've already conducted preclinical studies in uveitis. This is a systemic piece that I'm showing you here with our lead domain, which targets ICOS ligand. And if you just glance towards the bars at the end of that graph there, you can see that the untreated is in the black, and our ELN21, which is our lead domain, is shown there in the grey. So we see protection over this horrible disease. And that protection from the clinical score is also reflected in the histopathology as well, which are the slides across the bottom. But what's really critical here is to avoid the idea of using a needle and going systemically. So let's see if we can get it through the eye. So we've already conducted some work looking at corneal penetration. As you can see here in the beautiful green images, a small molecule, which is just FITC alone. We've got an antibody on the right-hand side, which is labeled with FITC. It cannot get through the cornea, but our Sullamur domain, which is labeled with FITC, can. And what we can do is we can actually measure the antigen-specific binding of those Sullamur domains that have gone inside the eye as well. And that's what the graph below represents. So you can see there in the red and the blue, that's binding target in an ELISA, and that has been extracted from inside the eye, whereas an FC reformatted version, you can see along the bottom, simply didn't get in the eye in the first place. We also have a second lead product, which is an anti-TNF-alpha domain we call ELN22, and we've already shown equivalent potency to Humira in cell-based assays. The biggest difference there, we're only 9% of the size, which is fantastic. So we can go site-specifically with these domains. And we're already working with companies right now who have contact lens type delivery systems to concentrate our domains on the cornea for a non-invasive treatment. What I also wanted just to convey today is that although the core business focus is on eye disease and inflammatory disease, we're also working in the world of oncology because Sullamur domains have broad advantages here as well. One project that we're working on right now is to coat our Sullamur domains on the outside of nanoparticles, so it's all about targeted chemotherapy. We're working with Queen's University Belfast on that project. But I think another aspect which really does show the benefits of the domains is to create these Sullamur drug conjugates. You've got something very small that can penetrate into tumours. It can survive the harsh conjugation that is required from putting a warhead onto it. And what ALMAC brings is that site-specific conjugation, so you can control the drug-to-antibody ratio. And there's a direct correlation between that and efficacy. This is our pipeline at present. So our anti-ICOS ligand domain has been through 2 preclinical models. Our anti-TNF-alpha is just moving into preclinical models right now. Of course, we're starting with something that's only 11 kilodaltons, so it's going to get cleared from the body very rapidly. We know that. So what we've done is we've developed our own half-life extension biotool, which is called Endure, which is an albumin binder. So what we can do is we can fuse that to our solar domains to increase systemic exposure. It's been through 3 different models. We are working with Merck right now in bioprocessing, and that project really exemplifies the ability of these domains to bind cryptic or hidden epitopes. That's going really well right now. And of course, ALMAC that I've mentioned already in the world of oncology. What we've done is we've created an effective drug discovery pathway, if you like. So we have a dual platform approach. We do in fact immunize these animals very carefully, and don't think shark, think smaller dogfish, please. But what we know about nature, we've created in a test tube. We have 100 billion clone libraries that we can interrogate directly against different targets. And they're producing single-digit nanomolar hits straight off. We can reformat, but you're starting with something really small. So why not keep it small? You can make bifunctionals very easily by just putting a flexible linker between them. And of course, you can make these Sullamur drug conjugates for cancer. But if you need that systemic exposure, if you want to go for a chronic disease that requires a greater therapeutic window, then we can half-life extend them using our Endure across a bifunctional if necessary, or a classic FC. If you want ADCC, we can do that too. And that opens up the opportunity for site-specific delivery, which is the route that we're going down right now, but also systemic delivery to open up that larger anti-inflammatory market. The management team we have in place, we have Chris Jones, who's our chair. He's had extensive experience in life sciences. He's had senior roles as CEO and chair before and succeeded in securing considerable investment in companies. Myself, so I'm ex-biotech, ex-pharma, and now I've come back to biotech to set up our own company. We have Tim Morley, who brings that expertise in preclinical to clinical, because that's the point that we're at right now with our products. And Andy Porter, who's had success in spinning out a company from scratch and having it acquired for a very large pharma company. So as I said at the start, we're currently looking for Series A funding of 10 million. That would be de-risked from SIB up to the tune of about 2 million. What does that get you? Well, that gets you our 2 lead products into the clinic, the ELN21 and the ELN22, through to phase 1/2A. What we'd also be able to do is to progress our oncology projects, particularly the Sullamur drug conjugates with ALMAC, and also an internal project that we've got at the moment. It will let us open up the opportunity for other inflammatory diseases with ELN21 and 22, and it will also let us do more exploratory projects, which is quite exciting, high risk, but very high reward, look at targeting very challenging molecules such as GPCRs and ion channels. And of course, our Endure domain, it's ready to license right now, and we believe that could be the first Sullamur domain in patients. So as I said before, just to reiterate, that will take our 2 lead candidates through to the clinic, also progress the oncology, the cryptic epitope piece, which I think is a very exciting area, and we fully believe our Sullamur domains can do that. Of course, we need to know what's happening and what's needed in the clinic, so we need a clinical director to make sure that we are always on track for that. And we've already managed to secure collaborations, access to platform, and joint benefits downstream from different partners, and of course, licensing Endure. So I hope what I've done today is let you understand a bit more about what a Sullamur domain is, that we're targeting a £3 billion market in the first instance, but that's a gateway opportunity to a much larger market. We have a great management team, a fantastic science team, a strong IP position, which is something that we've taken our time to ensure so that we can protect our drug development all the way through to the clinic. We're already working with some great commercial partners, and we're opening up the opportunity for multiple licensing opportunities in the future. I'll leave it at that, and I look forward to speaking to you afterwards. Thank you very much.
Any questions for now for Caroline?
Hi.
Hi. Is there something about the way sharks sort of mount their immune response that's different to, say, like camelids or
Yes.
that makes a sort of different sort of drug?
Yeah, well, I mean, they do. I mean, this has come from a comparative immunology piece. It was a discovery, like Ablinks. You know, they kind of looked on a gel and saw something interesting because it was a single domain. Similar to that, I mean, sharks have IgM, but they've got monomeric IgM, interestingly, which is adaptive. And this IgNAR, which is the origin of the VNAR, seems to kick off just after the monomeric IgM. But it doesn't look like an antibody, okay? So it doesn't have a CDR2, for example. It's got an HV2 and an HV4 region, which adds that level of diversity. We're not really sure how it's happened. I think it's just the benefit of gene splicing and such like, that these domains happen to have been part of the adaptive or become part of the adaptive immune system. But, you know, sharks have been around for 450 million years. They're doing something right. So I think their adaptive immune system's evidently pretty effective.